Hi, welcome to Project Spidey Mesocosm, presented by Isabel Logan, Pranesia Woldridge, Derek Sims, Stacy Ikarike, Faith Morrison, and Ivan Robles. We are 6 IB ESS students under the leadership of Mr. Random Hope. Project Spidey Mesocosm is a terrestrial ecosystem model named after our 2 fishing spiders.
And here's our introduction. Mesocosms are enclosed environments that allow a small part of a natural environment to be observed under controlled conditions. For example, containers could be set up with and without worms to investigate the effects of worms on terrestrial ecosystems. Another possible use of mesocosms is to test what types of ecosystems are sustainable. This involves sealing up a community or organisms together with air and soil or water inside a container. To summarize, mesocosms is any outdoor experimental system that examines the...
Hello there. For the Mesocosm project, we chose to build a terrestrial mesocosm. We used a glass beverage dispenser with a corkscrew top to build our mesocosm in. We used small pebbles provided to us by Mr. Pulp as the bottom of our mesocosm structure, as a drainage layer since the mesocosm was being built in a glass jar with no drainage holes. Next,
First, we have our sun's energy, and that energy flows into our brassica rapa, which is our producer. Next up, unfortunately, we did not have a primary consumer, but if we did, then that primary consumer would be consuming the brassica rapa. Next, we have our fishing spider, which is our secondary consumer. And once again, hypothetically, if we did have a primary consumer, then that fishing spider would be eating the primary consumer and consuming the energy from those previous levels. Next, finishing it off, we have our decomposer.
This is the diagram part of our mesocosm, where I am going to list the biotic and abiotic factors. For the biotic factors, there's the leaves found in North America, Arlington, Texas, the twigs and branches found in Arlington, North Texas. There's the 6 worms, 1 spider, let's see, what else was there? The moss, the oak leaves, mold on the cork, which is a bit hard to see, and the black locust leaves. And that's about it for the biotic factors. For the abiotic factors, there's the soil, which is actually designed to help plants grow, clay stilt, which was found here in Arlington, North Texas, North Arlington of Texas. The rocks, which are also found here in Arlington, Texas, and some subland rocks.
Our mesocosm was created on October 26, 2022. And our products inside contain Miracle-Gro all-purpose soil, some silty soil from the school compound, moss, brassica rapa seeds, sedimentary rock found in the school, oak twigs found outside in the school, and some lawn grass from the school as well. Some oak leaves, we got 1 fishing spider from outside, 6 redworms, black lotus leaves, and tap water from the school. As you can see, our data, this is the soil pH collection. Over time, it would go up and then go down based on the days we checked it. And as the temperature collection, we use Celsius. And in the beginning, on the November 1st, it was 23 Celsius. And then by the end, December 1st, it was 20 Celsius. So it reduced over time. And then our plant height, we measured in centimeters, and on the first day it was 0 centimeters, and then on the last day it wilted, unfortunately, and it became 2 centimeters. For our qualitative data, we showed the decomposition rate for everything inside the mesocosm and then the status of them. On the first day, the oak twigs and the branches and the leaves, everything was fine, and then by the end of the project, everything had wilted away. And then for our graphs, the data regarding the soil pH, our data changed over time with the positive increase. And then regarding the temperature collection, our data changed over time with an increase, but then a sudden decrease. And our seedling began to fall on November 24th, which was day 30. Here are our graphs. And that is it for the data collection.
In conclusion, we found that our mesocosm was not self-sustainable at all, but we expected this. Our Brassica rapa seedlings did grow, and they were growing pretty well, and they looked healthy up into our 8th plant height, which was about 6 centimeters. We suspect this is due to the pH soil levels changing and being unsuitable for the Brassica rapa, and also a lack of sunlight during Thanksgiving break, and probably the weather conditions as well. Our earthworms and fishing spiders also died within the first week. Rest in peace. And pretty much everything that did not have a root attached to it was already dying. But one thing our mesocosm did well was preservation. Our twigs, branch, and locust leaves were perfectly intact. There was no color changing or anything, but they did become decomposing and browning after the 5th and 6th observations. And then, as of yesterday, our locust leaves have not browned, and no organism has fully decomposed. Everything's there, dead but not decomposed. Well, not fully decomposed. So our limitations include a lack of resources, such as erosion, diversity, and a lack of an oxygen cycle. And these limitations probably led to our mesocosm not being self-sustainable. And although limitations like a lack of erosion and weather cannot be easily replicated in a mesocosm, we could have improved our lack of an oxygen cycle, species diversity, and resources, which would have saved our mesocosm, and our organisms would have likely still been alive. So this experiment was a great learning experience, and I think most of us would say that we would create another mesocosm again, but we would have to replace our 2 spiders, Dagmar and Deathblade. That's it, and here are references. Thank you so much, everyone. I hope you all have a wonderful day.